right, I think that works. So, hello everybody. Thank you for staying to the end. I timed this talk at 8 minutes, so I'll have you out in lots of time for the poster session. My name is Morgan Taschuk. I'm from the Ontario Institute for Cancer Research, and today I'm going to talk to you about MISO, which stands for Managing Information for Sequencing Operations. So what is MISO? It's a laboratory information management system specifically designed for technicians, like you see here, in sequencing laboratories. It records information about lab process, including sample preparation and processing, quality control checks, and measures kits used in instrument runs. If you've worked in a lab, you've probably used either a LIMS system or an LIS system. What we really want out of systems like this is their data. We want to be able to do analysis on it, to have it inform our future analysis, to help us plan our experiments, and to let us trace when things go wrong. We really want to be able to query it and mine it and come up with detailed insights about how projects and people work. And really our entire world, all of our beautiful figures and data is based on data like this, and so much of it comes down to all of this data entry in this LIMS system. So because what MISO actually is data entry. It is explaining to a computer in painful, minute detail what it is that you've done. Everybody hates it. Salespeople hate it, doctors hate it, researchers especially hate it. It takes too long. You can see on the zombie on the left says it takes too long. The gentleman in the middle says that it's repetitive and seems pointless, and the lady on the right says it's frustrating and takes time away from the activities she actually wants to do. Doctors hate them. I found this article in the New Yorker with this quote in it: "People in the medical profession actively, viscerally, volubly hate their computers." For them, data entry takes time away from their patients. One study found that during a patient visit, a doctor spends a quarter of their time during the visit on the computer, and then 2 hours of documentation for every 1 hour with patients. Every time we have to stop to tell a computer what it is we're doing, we're not doing that thing, and so it feels redundant and sometimes pointless. And often the software you have to use is cumbersome, and it has a considerable learning curve. And it's not just the users who have this misery, it's also the people who are using it who have the misery. They're data scientists and analysts whose work depends on the entered data somehow. Data scientists spend 60% of their time cleaning data, and they kind of hate it. If you work in data science, you've probably been a data wrangler at some point. You were given data, and you were told, make it ready. And so you probably bemoan the whole process and the unbelievable number of errors and duplications and omissions, and you're wondering, how could anybody come up with these many errors? And so how LIMS systems usually work is that you have the poor laboratory technicians who are doing their very best to enter things into their LIMS system, and you have the bioinformaticians who are just frustrated at the quality of data that's coming out the other side. So my group is on the right. We use the information that's entered in the LIMS in order to automate analysis. We are highly invested in making sure that people in the lab do it in a timely and accurate way, but it doesn't usually work out that way, and so we spend a considerable amount of time fixing mistakes. So at my institution in 2015, a miracle happened. My boss decided to replace our commercial LIMS system with an open source LIMS system that we, as a team of software developers and data consumers, could customize to our heart's content. That was MISO. It's an open source LIMS that was first developed by the Earlham Institute and Rob Davey in the UK, and since 2015, we've been the primary developers on it. So the first step was to figure out what was so wrong and so bad with the old system. It was hard to understand, it was difficult to use, and it was completely inflexible. Technicians had created their personal best practices that were undocumented, and they were passed down like a stinking inheritance from person to person throughout the lab. It was ritualized. If you clicked here and then you clicked there, something happened, and there was no reason why you had to do it that way. It used terminology that the lab didn't use, and it called everything a template, regardless of whether it was a template or not. It had no concept of a project, which is crazy. Somehow it was used for 7 years in our lab. So let's dig a little bit deeper. Why does this happen? Why does commercially developed software utterly fail at its only job, which is letting us enter data? This is not a new problem. This is a paper from 45 years ago when Rob Kling explained that some of these problems happened because we made our software for machines, not for people. So machine-centered software doesn't tolerate human error. It's made to be precise and require that precision of its users. The environment is set up to simplify the machine's processing instead of the user's processing, and the users have no say in their final design. But we're human, and we're not perfect, and we're especially not at our best when we are hating on the process. So no matter how you penalize the people who enter data, make them do it twice, make somebody look over their shoulder, they're still going to make mistakes because they're human, they're fallible. Autocorrect is a great example of this. So when it messes up, it's hilarious and sometimes really embarrassing, but most of the time it does a pretty good job of catching our mistakes, and a tiny bit of validation work can catch most of the significant errors. So in user-centered designs, people are accepted as error-prone. The burden of precision is placed on the machine. Designs and assumptions are intelligible to users. So my question when we started development is, how can we help our users succeed? They're highly trained, intelligent human beings, and they're doing the best they can under their trying circumstances. We want to give them all the tools that we can in order to get them to succeed. So our aim with MISO, we had two simple goals. One, it was to get out beautifully clean data and make data entry not suck. And equally important was to make data entry not suck. We're not perfect at this point. We're still working on things, but our focus is on making it better. So the first thing we do is that MISO uses the language of technicians. We want to minimize the amount of translation between what they do in the lab and what they actually have to enter into MISO. And to do this, we've actually asked our users what terms they use. We're lucky at OICR where we have a sequencing center right in the Institute. You might think this photo is a stock photo, but it's not. This is one of our technicians at OICR, and last year she won the prize for most tickets submitted to MISO. So she is one of our most reliable people that we can ask, no, what do you really do? And she'll tell us and help us inform and make it better. So you can see this is one of our menus. We call things projects and samples and libraries because that's what the lab calls them. We also have a really detailed sample hierarchy to capture lab operations. So a sample hierarchy describes all of the procedures that it takes for a sample to move from the identity to the tissue, all of the different ways you can process the tissue, all the different ways you can extract the DNA, and then going down all the different kinds of libraries we can produce and all of the different kinds of dilutions and aliquoting that we can do on libraries. So we're capturing the process in the technician's own words. To actually do the data entry, before, our users often prepared their sample sheets in Excel. Putting aside the well-known problems of using Excel for database functions like having dates or things that aren't dates spontaneously render as dates, we wanted to reduce the amount of context switching. So we didn't want them to have to tab out of MISO to go to Excel and do that multiple times. So that's a time waster. So instead, we built our systems to work like Excel. We wanted it to be usable without Excel, but have a familiar interface to reduce the amount of mental disconnect. So it has things like copy down and other functions, and it also does very specific validation. So on things that have to be in nanomolar, it actually says nanomolar on there, and it makes sure that you're entering a sensible number into that field, which is things that we can't really do in Excel. Well, you could do it probably with a macro, but we're not in the business of programming from Excel. In fact, I really tried to get rid of Excel in our lab, but I didn't actually fully succeed in killing it off. So there are still buttons to import and export to and from Excel. So you can do more complicated functions like summing, additions, subtraction, that kind of thing, and do that in Excel and then re-import it if you have to, and some people apparently do. It's okay. I'm over it. You might have noticed on the previous page, or in a couple pages back, that we have control vocabulary for consistent data entry. So we have these drop-downs. They are constrained while you're doing the data entry, but you can add it from going to portion of the interface. So this keeps the data entry consistent so that people can't introduce new terminology while they're doing data entry, but still permits things to change as it happens at the breakneck pace of research. We also have a number of built-in tools to help with good data entry. So one of them is the index distance tool. So since we got the NovaSeq sequencing instrument last year, all of a sudden we're throwing all of our libraries on a single lane of sequencing. And sometimes this means, especially if they were prepared with different kits, that there's a potential for their indexes to overlap. So we have tools built in to tell people in the lab whether or not their indexes are going to impact sequencing. It's also compatible with other software. So MISO is just for technicians, so we made sure that we can have it interact politely with a lot of different systems. So one of them is Gitter, that's our bug tracker system, and you can pull tickets from Gitter into MISO so you can see if there are any issues with the sample or the project that you're looking at. Prometheus lets us monitor activity on the LIMS anonymously so that we can hopefully make things better. Run Scanner and Pinery I'm going to talk about on the following slides. So Run Scanner is, it monitors directories on the system containing the output from sequencing instruments. So both for Illumina runs and for Oxford Nanopore runs and for PacBio, the RS2 runs, we don't have the SQL support yet. But if they're on disk and the software can read them, we can pull back stats about them. This is served as JSON through a REST API. And MISO uses this, and it pulls information straight from Run Scanner into its interface, and then it can do things like tell you the Q30 of a sequencing run in MISO, so you can kind of follow it in real time without having to log on to SBS. And the other tool that I wanted to talk about that it overlaps with is the Pinery API. So Pinery API is a RESTful interface for pulling back LIMS data. We're on the computational biology side. We want to pull back information from the LIMS in order to parameterize our analysis and also automate our analysis. We also create reports, and we don't want to have to go and click around the MISO interface and download an Excel spreadsheet for that. So we have the Pinery API, and we can make all kinds of queries and get back all the data that we could possibly need from the interface. We also have a complete user manual, Gitter chat, and bug reports here at this URL. So this is our short feature list. I just went through it, so I'm not going to read it again. Time permitting, I'm going to show you a quick live demo, and I'm not going to go too overboard here, so I can't actually see it here, so this should be interesting. Okay, so this is the MISO interface. This is just running on Azure. It's a tiny little toy box, but I preloaded it with some data. So if I go up to projects here, that says projects, right? I really can't see from this angle. So I preloaded this. This is a test project. So you can see at the top there's some basic information about the project that's related to sequencing centers. For example, the reference genome, so that we could pull that in later. Then down here you have all the samples that are associated with the project. You can see here that they are single-cell samples, and you can also see which tissues they came from. I'm leaning really close to the mic. Yeah, and then they come from different identities down here, so you can see all of the different sample classes that are part of the sample hierarchy. I also made libraries from these single-cell, so you can see all the libraries here, and I don't think I made anything else from that one. But you can see library templates and dilutions and pools, and so it's all available under the project page. You can also go list it under the samples page. And then if I can, did I click on that? No. There we go, I got it. I'm gonna just do a few because I can't see. And then I'm gonna hit edit. All right, so this is the Excel-like interface that I was talking about. I'm just going to do it in the description field here so that you can see. Oops. Maybe I'll do it in the drop. Nope. I'm sorry, I really can't see. All right, so you see there's the little dot here, so you can do the Excel fill down kind of stuff. So I wanted to show you some of those bulk functions. Yeah, and there's other things that I haven't mentioned. So obviously there's authentication. This one is just behind HTTP, but we have support for HTTPS. There is the support for actually ordering sequencing through this interface, and people will see the orders pop up, prioritization, all the tools along the side there. We have nice freezer maps so that you can actually see where your freezers are located and then go find your sample, and then you can make a little diagram of how your freezer's laid out and actually find where your sample is in physical space. Yeah, so that's MISO. I'm going to try and get back to, nope. Oh. I just have my acknowledgement slides, so we'll be pretty quick here. All right, so the last thing I want to say is what's next for MISO. We're doing a 1.0 release this year. One of the most exciting things that we're doing is that our lab is becoming CAP accredited, so it'll be able to process clinical samples, so we want to make sure MISO is ready for that. And so I just want to say thanks to our users. This is the genomics team. And also thank you to the MISO, oh, sorry. Did not realize that it wasn't showing. All right. Yeah, so MISO's been under development for 9 years, and now we're planning a 1.0 release. So look forward to that later this year. So thanks to our users and to the full MISO team. And you can try it yourself. So that demo instance that I just showed there now has its username and password freely available on the internet. So we'll see how long I can keep it up before somebody takes it down. Yeah, feel free. I'm also going to be at the poster at P42 right after this. So if you want to join me walking over there and we can talk about MISO, that would be great. Thank you. Thank you.
Okay, so are there any questions, comments? It works, yeah. Okay.
Yes, briefly. A user in your scenario, is that a member of the core facility of the sequencing team, or is it also a request form, like where anyone who ever wants to submit a sample to the facility can enter a request?
It's technicians, yeah.
It's technicians only, so that's the, yeah.
Yeah, we do have plans to build a system that will essentially be able to start a project in MISO in order to say how many samples they're going to send and to be able to do a sample submission form. But it will be separate from MISO. MISO is for the technicians.
So in that context, if you ever want to collect user sample, because so many things happen upstream of the actual sequencing project, so what we find very difficult is now retrospectively collect the metadata of the samples. So yeah. I'm not quite sure whether this is not a MISO-related question, but whether this system is extendable to also collect metadata about the samples, yes. Gender and all these things.
So sex is one of the options that you can fill in. So you might not have been able to see it on the quick slide through, but we have a lot of different data that we collect. Part of that is just being able to automate analysis. We need to know whether or not something is a tumor or a reference because we're a cancer institute. So we do collect a lot of things. We collect sex. We collect secondary identifiers, which is what would the institution that sent us the sample call it? And then once we have that, we can start doing all that kind of that linkage. But a lot of these fields are actually required in our system, and we can make them required for particular projects. There's one project in particular that requires sex to be on the list. So yes, we do collect a lot of that.
Hi, Megan. I have two questions. I called you Megan Morgan. I have two questions. Can you talk a little bit about the back end? Is there some database on Azure? And two, kind of how many people are working on it now? I'm sure there's a lot of development going up to this point, and then just kind of, yeah, how many people, how many developers?
Sure. If I could get back to the acknowledgements. Oh, there we go. Yes, so the back end is a MySQL database with a Hibernate and Spring connection, running a Java application that is has a JSP front end, and we serve it over Tomcat, so that's the back end. There is one full-time developer right now, and we have the MISO team that's there. Dylan Cook is our primary developer. Heather Armstrong, Andre Masella, and Alexis Horsava all kind of step in as necessary. Yeah, but one full-time developer.
So I liked your comment on how, how a lot of the value of this system comes from customizing it to really fit what the technicians are expecting and using their vocabulary and really focusing on them.
Yep.
But of course, this also comes at the loss of some generality and some flexibility. So I'm wondering how difficult it would be to adapt MISO to a different laboratory, maybe even something like functional genomics, or how far from your specific use case do you think MISO would still be useful?
I would say that at this point, if it's an organism that is a eukaryotic organism where we can reliably sequence a single individual, MISO is set up for that. So I would say mice, humans, maybe even C. elegans if you're doing particular individuals. Once you get down to yeast kind of sequencing, I don't think it would qualify much anymore, because it really depends on that identity, which is linked to a specific individual. As for customizing for a particular lab, we have so many different sequencing projects that come in with their specific protocol, and we actually add in new sample classes and remove them all the time. So one of the things in 1.0 we want to do is make that really easily customizable through the interface. And so we're hoping that as long as you do some kind of individual sequencing, not bulk, not batch sequencing, that MISO should work for you.
But it would be a major effort to adapt this to a mass spec facility or something else that has similar workflows.
Yeah, it's predominantly for sequencing operations. We do have support for microarrays, limited, but not for mass spec at this time. Yeah.
Last question.
Yeah, I had essentially the same question. So do you know, is anyone else using MISO outside of your institute?
So it was originally developed at the Earlham Institute in the UK. And they are, as far as I know, they're no longer using it. They moved on to some commercial option. But we have a number, one of the things about being open source is that anybody can clone it and use it. So all we have are bug reports that come in. So we have at least one individual in Berlin. I believe his lab is using it because we get constant bug reports from him. And there are 2 or 3 other people scattered around the world. If you go to our issues on GitHub, I love issues, by the way, if you go look at bug reports, because it tells us when people are using our software. There are several other individuals who are filing issues, which to me says they're probably using it.
Okay, so thank you very much, Morgan. Thanks very much to all our other speakers this afternoon, and hope you enjoyed the poster session.